Good afternoon or good morning, everybody, and welcome to our third webinar in this series from the British Society for Immunology. I'm Doug Brown, Chief Exec of the British Society for Immunology, and thank you very much for joining us. We have over 1,000 people again viewing this webinar live, which is absolutely fantastic, and again with a global audience. In fact, we've been completely overwhelmed with the response to these webinars, which is why we're now delivering these in association with the British Society for Immunology's official journals, Immunology and Clinical Experimental Immunology. And we've been very fortunate to receive some much needed financial support from the Lorna and UT Chernijovsky Biomedical Research Foundation, of which we are very grateful, and is making our coronavirus work and these webinars possible for the coming weeks and months. So thank you very much to those people. We are continuing our coronavirus work, and I encourage you to visit our website. We have updated it with new resources, new information to support you and your colleagues at this really challenging time. So please do take a look at that web page on our website. But now it is my pleasure to introduce our speaker for today, Professor Danny Altmann. You may recognize him from our webinar last time, but Danny is here again to give us a fantastic overview of some of the emerging research in COVID-19. Logistically, Danny will speak again for another 30 minutes or so, and then we will have time, 10-15 minutes for questions at the end. So please do submit your questions via the question functionality in the toolbar of GoToWebinar, and we will ask as many of those questions as we can of Danny at the end. So now it's time to hear from Danny. So Danny, it is over to you.
Good. So welcome to the third of these BSI COVID-19 webinars. As I mentioned last week, in our lab, going back to 1990, actually, we've every week done a journals club where we try and dissect and analyze a recent paper and get them on board. So in that vein, I guess like lots of people, I've been over the last few weeks continuing to read every possible paper I can of those coming out about COVID-19 immunology. So today I wanted to go through some of my favorites from the last few weeks as a way of addressing some of the key issues. If I try and so some of the points that that I think have been on our minds that are addressed by recent papers are these. So can asymptomatic people shed virus, and if they can, what are the implications of that for case identification and transmission and blocking transmission? The thorny issue that I think plagues all of us at the moment, antibody testing, achieving herd immunity. What exactly do we need to know, and how are we going to assess how much immunity is enough and when we've got there? Do we need neutralising antibody tests? Some discussion about that. Moving on to some slightly more in-depth discussion about therapeutic neutralising antibodies in their various different forms and how they could be applied. And then we'll move from there to talking about more broadly about immune therapeutics, things like anti-IL-6 and anti-TNF trials, and then a little update on vaccines. So to start off, this is a paper that came out yesterday in Nature Medicine that I thought was illuminating. So what they've done is they've looked at transmission pairs and used that to build a mathematical model. And you'll see here that what their model predicts is that you can assume that people were infectious for 2.3 days prior to showing any symptoms. And if you use that for the basis for your modeling, the answer that you come up with is that 44% of all transmissions that occur during this pre-symptomatic phase. So in other words, people out in the community spreading virus without knowing they're ill and without knowing they're doing it, which obviously has quite profound implications for our policy and control measures. But if we go So I'm just trying to move on the slides. Yeah, so if we go back to some of the key immunology questions that are on all of our minds at the moment, some of the things I've been trying to think quite hard about are these. Is it a given? Is it a fair assumption that all those who've been exposed to the virus will have made an antibody response? That's something we touched on last week. If the tests that we're looking at are about detecting virus-binding antibodies, whether in a sort of lab-based ELISA or in some kind of lateral flow commercial module, is this precisely the same as having functional neutralizing antibodies that protect against infectious virus in any second wave? And then we speak very loosely about protection by herd immunity. What is it? How much is enough? And as you'll see, I think the calculation matters and depends on whether we've really understood this R0 value correctly. So let's start with the question of herd immunity. So we've all heard, oh, expressions in the newspapers about things, with phrases like, we'll let it rip through the community until herd immunity develops. So if you put to one side what you think of the cost of ripping through communities, what is herd immunity and how will we know if it's developed? So herd immunity is a rather specific immunological parameter. You can model it, you can calculate it. So there's a fairly key historic paper here from Roy Anderson and Robert May in Nature that lays out a calculation for it. But the key point is you can't guess it, and you wouldn't guess it. You wouldn't, I don't know, you wouldn't set out on a transatlantic airline and guess how much fuel you needed to get to the other side. You'd calculate it. And herd immunity is in that kind of category. You need to know it. Knowing it is of value. This is a model from a paper about measles, obviously one of the most infectious diseases we know of, and it's in Lancet Infectious Diseases. I've given the full citation here. And the reason I think we need to be familiar with graphs like this is that they matter for our calculations and our policy, because if you look at the shape of the curve, if we think that our baseline R0 for this disease is, as came out of some of the Wuhan calculations at the beginning, about 2.2 to 2.5, you're over here on the curve, and you need something like 60% herd immunity to be safe. I know that some people and some models and some assessments are now saying that that was an underestimate, and our calculations need to be somewhere up here around 5 or 5.5. If those estimates are right, then our herd immunity doesn't need to be 60%, it needs to be nearer 80%. Okay, so these are finite models and calculations that matter. So if we're heading for 60 or 80%, 60 or 80% of what immunity do we need? And this bears on something that all immunologists deal with and think about, the kind of concept of correlates of protection, a term that, as far as I know, came from papers by Stan Plotkin over the last several decades. And these are some quotes from one of his key papers from Clinical Infectious Diseases many years ago. So he says some quite useful things. Correlates of protection after vaccination, but obviously it could be natural infection, are sometimes absolute quantities, but often a relative, such that most infections are prevented at a given level of response, but some might occur above that level because of either facing a large challenge dose or having some kind of immune deficiency. And there may be greater than one correlate of protection for a disease, co-correlates. These might be in different parts of the immune repertoire, effector cells or memory cells correlating with protection. It may involve a different range of T cell or B cell effector functions as co-correlates. And often, unless very specific studies have been done, we might not know all the true functional correlates of protection. So we use surrogate tests, often antibody measurements, as a kind of biomarker of these. So what do they look like? This is important, yeah. So this is from Stan Plotkin's paper in 2008 for a number of different infectious diseases. And if you do the data analysis on this slide, you can see immediately there are different infections, different tests that are used, and different correlates of protection, which are quantitative numbers that matter. So, for example, they matter so much that they're used for licensing vaccines in terms of whether they can attain the efficacy that you need to block transmission of that infection. So it goes without saying that for the infection we're talking about today, unless I'm corrected by anybody, we haven't the foggiest notion of what those values are, and we do need to know. So how far are we off our 60% or perhaps 80% herd immunity, depending on our assumptions of R0? So I talked about this last week. There's lots of numbers beginning to come out from people doing serological testing, sort of here and there. I haven't seen any very large or comprehensive data sets published yet, so you'll forgive me if I'm taking someone off Twitter, but there's a Dutch data set here from blood donors talking about 3%. There's Florian Kramer, who's done a few thousand in New York, talking about up to 7%. There's a Scottish blood donor survey talking about 0.6%. So I think that by anybody's estimates, our herd immunity is a considerable distance off those 60 to 80% estimates, which, to state the obvious, means that natural exposure probably isn't going to get us there, and we are going to be dependent on vaccines. So obviously antibodies come in many different flavors with many different effector functions. So let's do a brief reminder about virus neutralizing antibodies and why we care about them. So quite a good way to think about them in this context is to go back to papers on the related virus on SARS infections from a few years back. And these are two different papers looking at neutralizing antibody titers. And they both, one comes from Robin Weiss's lab and one comes from Zhao Ningzhu's lab, but they both involve the kind of Robin Weiss toolkit using pseudotype virus as a kind of lab safe way of making a mock-up of the antigens of the infectious virus in a safe virus, so that you can do a sort of model study of neutralization of antibody entry. And Robin has really been the master of those kind of assays for probably about 50 years. And so in the first paper here on SARS patients, he says that all patients were positive for neutralizing antibodies, and they analyzed them for greater than 200 days and they persisted. So that sounds like good news. And in a kind of follow-up related study later on, right here at the bottom of the page, he says that in a SARS cohort, 90% of them had strongly neutralizing antibodies. So that sounds like really good news, doesn't it? But obviously there's a kind of caveat there, because these are studies of people who've been severely enough affected to be hospitalized, whereas for the present discussion, we're not talking about that. We're talking about the whole community, including people who have asymptomatic infections and barely knew they were infected. So what do we know about the antibodies that they might have? So lots of labs and lots of preprints, lots of papers have been looking at that in the context of different ways to set up antibody assays for SARS-CoV-2. This is one that I like from groups in Holland and Germany and France. And in one of the tables from their paper, they address the issue that many people have been talking about, the sort of potential confounder for antibody testing, that SARS is part of a family of coronaviruses, and it has near neighbors and distant neighbors. And how much of a worry would it be that we could pick up false positives from people who have antibodies to the other coronaviruses? And if you look at the sequence homologies here that they've worked out for the different antigens that have been used in tests, including mainly the different versions of the spike antigen, you can see that probably it's not an enormous concern, because the nearest homology is not that great. In their analysis, that's with SARS and then MERS, but not that many of the global population have pre-existing antibodies to SARS and MERS. And the common cold coronaviruses are really quite distant. So as you'll see in a minute, I think that's the kind of, in theory, worry that doesn't really take off as a major confounder, because some of the data is here. So on the bottom right, you can see that data about the confounder from the other infections, and they're not really giving you a big false positive signal. all the signal is from their people who really have had SARS-CoV-2 infection. And then they give you a little taste of their data here with severe or mild disease patients. And that's the same kind of answer as we talked about last week, that people have very variable responses, some of them kind of negligible. And the two groups of people who I think have those negligible responses are either the very sick ones who might be at risk of dying, or the very mild ones who've been very marginally affected, have seen a very low dose of virus and therefore a very low dose of antigen challenge. So really, may not have detectable protective neutralizing antibody on board. So that's the take-home messages from their study. Many others. So one that I talked about last week, this one from Florian Kramer's lab that he circulated to labs around the world, and the route for their lab and their collaborators in the US has now been to give authorization to roll out that test via the FDA, which will now be rolled out as a lab-based test very widely, which I think is a terrific development. Or this study here from Shanghai that I like, which uses the kind of test that Robin Weiss had talked about, looking at real-life neutralizing antibody titers in a cell-based assay. And again, I think the take-home message from that is very encouraging, isn't it? That the absolute vast majority of the people they look at have not just antibody titers against the virus, they have neutralizing antibody titers against the virus. And one of the worries that we had, that immune senescence is such that old people are very vulnerable, and old people certainly have different features in their immune response and might not make a good protective antibody response. Here, the 60 to 85-year-olds, if anything, have the best response, possibly because they had the biggest antigen load on board, and they look, kind of protected. So are there grounds for optimism that the majority of people who've had symptomatic disease, perhaps even fairly mild disease, have antibodies on board, and at least part of this is likely to be neutralizing? So it looks like people have made antibody, a part of their response is neutralizing. So this audience will be very well aware that the last decade or two has been an enormous time of growth for research on human monoclonal antibodies, and particularly neutralizing monoclonal antibodies in infectious disease. And they can be utilized in various different ways, but a fantastic growth area in immunology has been the very fine mapping of those and the analysis of neutralizing epitopes, and then making them in large quantities and rolling them out for therapeutic applications, for example, to block viral infection. So to talk about that for a few minutes, we can go from low tech to high tech, and that's not meant in a derogatory way. It's simply that the spectrum of applications that are out there. So people will have seen lots of talk about using passive transfer of immune plasma from convalescent patients and transferring that to sick patients, and that's been a sort of major direction of travel in the USA. And I guess it sort of builds on the kind of rationale that's very prevalent in the clinic for many years, of using kind of gamma globulin as a kind of catch-all therapeutic, but here in a more targeted way, using plasma from people who've got proven quantified neutralizing antibodies associated with their recovery. So a very large number of trials being rolled out on that, and a very big push from the FDA in the USA to recruit plasma donors for that therapy. And I think that's really plausible and interesting, and a really interesting space to watch. Or you could, if you wish to, go from low tech to high tech. So there are some labs in the world that have devoted the last several years to picking off important, challenging human infectious diseases and making enormous numbers of human monoclonal antibodies and throwing the full structural biology toolkit at them in terms of things like crystallography, affinity measurements, cryo-electron microscopy, to map at the finest structural detail the precise binding site between viral antigen and neutralizing antibody, and then feeding that information into therapeutic trials to try and optimize protection from live infection. So in this example here, it comes from James Crow's lab at Vanderbilt, and if you don't know this stuff, look up James Crow's papers, because he's done absolutely stupendous work on this across many infectious diseases for many years. And in this example from last month, they're looking at how you could make a cocktail of 2 neutralizing monoclonals against Ebola virus, and by the kind of complementarity between recognition of those epitopes, you can get really robust protection from lethal Ebola challenge. So if you can do that in this context for Ebola, is it science fiction to think that you can do it for really tough human infectious diseases? The answer is, there's a lot of data out there and a lot of clinical trials out there. So this is from a review article on the use of neutralizing monoclonals in HIV by Michael Nussensweig, who's been one of the movers in this area. So top right here is one of those graphs of FDA approvals for therapeutic use of monoclonals. Obviously, that's total monoclonals against things like cytokines and things as well, not just neutralizing monoclonals. But the number is very large. It's now up over 80 different therapeutic monoclonals in use. There've been enormous ongoing trials for their use in prevention of HIV, now used in a complementary therapy with antiretrovirals. So it's obviously demanding and expensive and non-trivial, but achievable over quite a rapid timeline, and definitely kind of out there and on the table. So many of you will have seen there've been a vast number of papers like that over the last month or so, with labs seeing the potential for doing this and rolling out different versions of their tried and tested neutralizing human monoclonals against SARS-CoV-2. This is one of the examples. There are many of them out there. So I think that's a really interesting and tractable and plausible space to watch, and I think the good stuff will come out of it. But meanwhile, obviously, there are lots of other therapeutic targets you'd want to think about for monoclonal antibodies. This comes from a paper last week in The Lancet by Mark Feldman and Tiny Maney and Tracy Hussle and others, and they point out that really high levels of serum TNF, tumor necrosis factor, are a biomarker of severe outcome. And that's interesting because obviously for more than 20 years, there's been a really big tried and tested track record for anti-TNF monoclonals, things like etanercept, where we know an immense amount about how they work, how well they work. Rheumatoid arthritis, the main application is an enormously common disease on planet Earth, perhaps 1%. So we know an immense amount about the efficacy and safety of these drugs, and we need to see trials for anti-TNF monoclonals in this disease. Anecdotally, there have been a few reports around about people on etanercept having done much better in the face of this infection than would have been expected. But obviously, we need to know far more than that in RCTs. I mentioned last week that high serum IL-6 is similarly a biomarker of severe outcome, and this is maybe one of the most interesting papers online about it at the moment. It's a meta-analysis of 8 different published studies, and you can scan down to the conclusion at the end that says that in patients with COVID-19, IL-6 is significantly elevated and associated with adverse outcomes. Inhibition of IL-6 with tocilizumab appears to be efficacious and safe in preliminary investigation. Results of several ongoing trials awaited. So I think that's a state of play, but I think there's some quite nice data out there. So lots of really doable trials like that. And this is a screen grab. Sorry, it's a bit blurry, but I've stolen it from somebody I don't know called Jesse Rafel, MD, and he's done a really good job on Twitter of collecting the data of clinicaltrials.gov of all the different clinical trials that are underway around the world for therapeutics for COVID-19. And you can scan down the list at your leisure, but in fact it's already out of date, because when I looked this morning, there were another 100 registered intervention trials, so now up to 14 different trials. And there's some really exciting, plausible approaches there. So, for example, one of the things that I flagged up last week was the fact that some of the immunology studies show the possible problem of immune exhaustion in virus-reactive T cells. And so there are 2 different trials underway to borrow from the oncology trials in immune checkpoint blockade and see the impact of that. But as you scan down the list, lots of other really, really exciting approaches. Lots of good stuff to look at. And then as we mentioned before, a big part of the exit strategy is the vaccines. And like you, I look at this quite a lot and quite often, and every time I look, there are new vaccine candidates being talked about. So this is quite a good review that appeared in Nature Reviews in Drug Discovery, and it comes from the team at CEPI. And I think last week when we had this conversation, we were talking about 60 or 70 known vaccine candidates in preparation. Now there are 115 being talked about. And even if you say, well, some of those might be, at rather early stages of development, many of them aren't. Many of them are in preclinical development or quite far down the line in, first immunizations in phase 1 trials. So, my view is that the candidates will, pan out a little bit differently. Some might look more efficacious and robust than others. Will almost certainly need more than one. So I certainly see this as a kind of, nationalistic, jingoistic race to the finishing line of one country's vaccine against another. I think we'll need a number, and many of them will make it to the finishing line with really good credentials. And you saw from the herd immunity figures that we really need them. So I'll end there, and like last time, I'm terribly happy to take any questions.
That's fantastic. Thank you, Danny. So we've had lots of questions coming in, as you can imagine, and we will probably start, I think, with one on the recent Gilead findings that remdesivir seems to have a beneficial effect on COVID-19 patients. Can you possibly comment on that?
Yes. So like everybody, I watch the antivirals with keen interest, but as a professor of immunology rather than virology, it's slightly out of my kind of core knowledge base. So I'm doing the same as everybody. I'm watching, in a sort of hard-nosed way to see what comes out of the randomized controlled trials. And I've seen some good stuff and some slightly less good stuff in terms of people's findings. But the simple answer is yes, I feel very optimistic. And I think it definitely will be part of the toolkit.
Great. Thank you. And there were quite a few questions that were coming in about concerns over the possibility of FC-mediated antibody enhancement of infection. Did you want to say a few words on that?
Yeah, so let me try and say some careful words. So the first thing to do is to state the obvious, that when I talk about the kind of race to the finishing line for the vaccines, although there's great urgency here, the safety testing, will be paramount and won't be relaxed. And, it's a well-trodden path in terms of safety assessment of vaccines. And for licensure, in any country, in any community, people will want to know that they're safe. And that'll mean that they're robust in terms of their efficacy, that there isn't some kind of damaging immune pathology, for example, from Th2 immunity, and that there isn't this business of antibody-dependent enhancement, i.e. enhanced uptake of virus into host cells. The bits of relevant data that I've seen up to now, I felt moderately cautiously optimistic that it wasn't going to be a big issue. I'm sure that somebody will come up with a study that proves me wrong, but I feel cautiously optimistic that it's not going to be a big confounder.
That's reassuring. Thank you, Danny. And there was a question that came in from, let me find it, came in from India. So thank you very much to our global colleagues joining us. That was a bit of crystal ball gazing and really a comment from you that in the coming years and decades, do you think it would be more useful to do high quality research into monoclonals or into vaccinology for managing future pandemics?
You know, I'd love both, but you know, again, just to state the obvious, you know, over the last many decades of vaccinology and efforts of bodies like CEPI and WHO and Gates, the aspiration has been to get global coverage at high throughput and low cost, for a number of a few dollars per person, and that's has to be so doable and such an important challenge, whereas having clinical grade monoclonal antibodies is always going to be a kind of high tech, high cost, emergency only undertaking.
Yeah, no, so both, we want both. That's what we'll be pushing for, so that's good.
One is more affordable and plausible than the other, in the short term.
Definitely. And we had a number of questions coming in looking at how different populations are disproportionately affected by COVID-19. So some questions around the biological evidence that can give us some insight into how it's disproportionately affecting Black and Asian communities, but also men who seem to be more severely affected by COVID-19 disease.
Yeah, so it's kind of thing you'd want to set a sort of medical student graduation essay on for their final exams, isn't it? Because you could really piece together a good point-by-point answer on this. So where would you start? You'd say, okay, men versus women is really interesting because we know so many parts of the immune response that are different between men and women in terms of the way that, all of the obvious, the way that sex hormones skew your immune subsets during development. An awful lot of those tend to come out with women more badly affected than men. If you think about things like any of the autoimmune diseases, quite a lot of them are female skewed. So you need to investigate a mechanism that can explain to you why this one is male skewed. That's really important. And then you've got all of the other stuff about the general population, and you'd have to build up your your undergraduate point-by-point answer by saying, number one, different people have different exposures. So if some people are in demographics or jobs or geographical environments where they have lots of viral exposure to a very high dose, they're going to do far worse than people who have low exposure to a low dose. So that'll affect differences across different populations of people. Then there will be differences in comorbidities. So we know that hypertension and diabetes are much higher in some parts of the world and some ethnicities than other. That's going to be an enormous factor. Then there's going to be more nuanced things, like we know that there are polymorphisms in the receptor, in the ACE2 receptor in humans. Some of those will be distributed unequally across the populations. And then we know that there are innate immunity genes that will play a part here, things like the IFIT type 1 interferon pathways that we know are different. For example, high susceptibility versions, quite common in the Chinese population. That's going to be a factor. And then down and down and down, HLA polymorphisms, and how long have we got?
So quite a complex picture you've painted us there, but something that could definitely keep the research community busy for some time yet to understand that in some detail. Definitely. And there were some questions coming in about viral dose versus antibody titer. Are there any studies that are emerging that are looking at a correlation there, but also how viral dose correlates with the severity of disease?
So maybe we need another webinar on that. So I've seen some quite nice papers on this. So the gut feeling out there is that there's quite good correlation, isn't there? That why is it that there are people in the community who clearly get sick and get a little bit sick, and perhaps have a few days at home with severe headache and cough and then get better, and other people obviously get very, very severely sick. And sometimes it feels like healthcare workers, for example, are disproportionately, quite badly affected. And there's a gut feeling that that's to do with antigen dose, and to do with viral dose, and to do with repeated exposure. And I've seen some quite nice papers starting to look at that. And I think we need loads more studies looking at that, because there's a lot of relevant stuff in there that we really need to know about. And then, as the question suggested, that's the driver for higher antibody titers. But being a person who's got high antigen exposure and high antibody titer might not be the group you want to be in. So although you've got a lot of antibody on board, you've paid a very high price for it. And so you'd probably be you want to be, if you had a choice, it will either, safe and happy with no virus, or in the kind of moderate protective antibody group. Because, rip-roaring antibody titers are a sign that something really bad has happened to you.
Definitely. Thank you, Danny. And then a couple of questions about different molecular mechanisms that may or may not be important. It'd be good to get your view on the emerging research there. So is there a role for inflammasomes in the disease and the potential of targeting inflammasomes in the IL-1 family?
Definitely, yes. So again, we need some more journals clubs. So I've seen good papers on this, and so far, because we try to do quite sort of high-level webinars of a kind that address the debate that's going on, in all countries at the moment about lockdown and immunity and herd immunity and release of lockdown, we've done the discussion at quite a kind of, high level of immunity, seen through things like antibody titers. But like any infectious disease, there's a whole part of the story that's to do with the initial innate pathways, the initial innate recognition, the TLR recognition, the inflammasome activation, and so there's already been some really good studies on this, and I think some evidence I've seen emerging that, as you'd expect for such a successful virus, it does have some strategies for subverting innate recognition, and a really, really productive area for research. And
how about JAK inhibitors? Do you think they have a role in treatment of COVID-19?
I think they probably do. This morning I was scanning down that list of 400 clinical trials. I don't think I saw any JAK inhibitors on there, but I might be corrected by anybody who's doing it. But yes, I think they probably do.
And there was a question in about zinc and zinc ionophores as well, and probably more virology than immunology. But do you know anything around the emerging research there in inhibiting the replicase enzyme?
Oh, that's way outside my knowledge base, but I'd love to hear from anybody who knows about it.
Okay, I thought I'd try that one on, but that's fine, Danny. Completely understandable. Then we had a couple of questions in about testing specifically. One on the swab test, which was if somebody has a positive PCR but is completely asymptomatic, can they still transmit the virus to others?
Yeah. So. And that relates partly, obviously lots of good papers on this. It relates partly to that paper I showed at the beginning from Nature Medicine yesterday, where, I know for the purists, there's some discussion around whether the best always shows real life infectious live virus, or whether it could be showing residual RNA. But as a rule of thumb, if you're swab test positive, I think you're virus positive, and if you're virus positive, you're a potential shedder. And that paper said, that their model was that you were accounting for 44% of all transmission, or you and people like you. So it's a really, really hot topic, and the kind of thing we need our policymakers to understand. It's not just about people in hospitals; it's about all the other people out there.
No, absolutely. And with the array of different surveillance tests that look like they're emerging, what's the likelihood we'll end up with more than one standardized test for surveillance?
I think it's fairly likely, and certainly, obviously, as I've shown already, in the race, different countries are evolving different local answers, and I don't have a problem with that. So, the kind of thing I was showing from Stan Plotkin's paper, where in the fullness of time, the community through various WHO documents and things evolve some kind of consensus on a test of choice, and a neutralizing antibody titer of choice or whatever is way down the line. And at the moment, we're slightly in kind of desperate measures. And I think for different communities or health authorities to come up with tests that work, I think would be better than nothing and fine for everybody.
Great. And the final question before we let you go, Danny, is that, well, there were a number of questions coming in about any implications of this viral infection for blood donation in terms of antigens, antibodies that might be in the blood of those who are or who have been infected.
Yeah. So again, I don't want to give a sort of inaccurate answer here, because I know that, again, in lots of different countries, the blood banks will be thinking very hard about this. And you'll have seen that some of the data I showed had come out of starting to screen blood donors. So they will be thinking about it. They will want to know it. And the reason, I guess, that you'd want to know it is just the obvious, that in one of the papers I was showing last week from Wuhan, there was a proportion, quite a low proportion, but a significant proportion, I think it was something like 5 or 6% of people who had a viremia, who had live virus in blood, and you wouldn't want to be passing it on to people. So I'd love to see any answers or any data from the blood banks about how they're addressing it, but I know they are.
That's great. Well, thank you, Danny. Thank you again for a fantastic talk. As always, a great summary of the emerging research in these specific topic areas. And I'm sure we had 1,300 people viewing this live, and I'm sure every single one of you took a lot away from that presentation. So thank you, Danny. It's time to draw this webinar to a close now. Just to thank you all for joining us. Really great to have you here. Just to remind you, we have the 2 previous webinars. The recordings are on our website, on our coronavirus web pages. And the recording of this one will be up on that web page soon as well. So please do spread the word and share this with your colleagues who are not able to join us live today. We have more webinars to come, so please do, again, keep an eye on our website, look out for the emails from the British Society for Immunology, but also our Twitter account as well, handle BritSocIm. And don't forget that the British Society for Immunology is here to support you. As I mentioned before, it's a really tough and challenging time, whatever stage you are in your career. So please do visit our website and please do engage with us and let us know what more we can do to support you during this difficult time. So thank you again, everyone, for joining. Thank you again, Danny, for being such a wonderful speaker and giving up more of your time. And thank you to our supporters as well, our journals, Immunology and CEI, but also the Lorna and Yuti Chernijovsky Biomedical Research Foundation, who have made this possible. And we will see you next time.